In this problem-solving video, we will consider why consuming a high-fat diet for 48 hours results in increased blood-free fatty acid and decreased GLUT4 content on the skeletal muscle membrane. First, it is important to consider that consuming a high-fat diet will result in an increased blood-free fatty acid content. This makes sense since eating more fat will result in more stomach and intestinal fat absorption, leading to an increased inflow into the pool of free fatty acids in the blood. What we will now consider is why increased blood-free fatty acid results in reduced skeletal muscle GLUT4 content. So the mass that we're going to consider in our video is GLUT4. GLUT4 is basically just a protein that can be found on skeletal muscle that is used in glucose transport into the muscle. So what we saw in the individual that ate the high-fat diet is that GLUT4 was decreased. So this would come about as a result of either increased outflow or decreased inflow. Basically, the outflow from GLUT4 would be removal from their membrane, and inflow would be translocation of GLUT4 to the membrane. So based on what we learned in class, we think that inflow is going to be the more important factor, more important than outflow in the GLUT4 decrease.
So let's look at inflow a little bit more closely. As Drew said, the inflow to GLUT4 in the skeletal muscle membrane is GLUT4 translocation. So down here we've got a glucose storage vesicle, or a G4 storage vesicle. So how does this translocate to the membrane? Let's take a closer look at the steps that lead to this. First, we've got insulin, which is released into the blood. Insulin comes through and is accepted by the insulin receptor. When this happens, a series of events occurs, which leads to the translocation of GLUT4. First, IRS is activated. The activation of IRS leads to the activation of PI3K. The activation of PI3K leads to the activation of AKT, or protein kinase B. What we see is that PI3K and AKT play key roles in the translocation of GLUT4 to the skeletal muscle membrane. First, AKT phosphorylates AS160, and what this does is it deactivates it, which means that the GLUT4 storage vesicle can now translocate to the skeletal muscle membrane. As well, PI3K activates RAC, and what RAC does is it helps bring that vesicle to the membrane again by transporting it along the cytoskeleton. Alright, now that we've developed our beautiful model, we're going to consider how it works in terms of the problem we're trying to solve. So as we can see, if we have a high-fat diet, we're going to get lots of free fatty acids in our blood. This is going to increase the concentration gradient for facilitated diffusion via the protein CD36 into the skeletal muscle. When this happens, we get an accumulation of fatty acyl-CoA. And fatty acyl-CoA has three different fates in our muscle. The first it can be oxidized in the mitochondria to produce ATP, so it disappears. The second, it can be reesterified, so it can be stored as fats. Or the third is sphingolipids can be formed. And when considering the problem, we're really going to focus on the effect of sphingolipids. So what happened is that fatty acyl-CoA turns into sphingolipids such as ceramides. And when this occurs, this isn't good for the muscle. Because what happens is that ceramides are known to inhibit AKT or PKB. And so again, what we see when this happens is that we get less deactivation of AS160, and which results in less inflow to the GLUT4 protein on our membrane. So our hypothesis is that muscle membrane GLUT4 content is decreased during a high-fat diet because resulting increases in ceramide impair insulin-mediated AKT phosphorylation.
Similar to the hypothesis we just presented, a study conducted by Strakowski and colleagues examined the relationship between insulin sensitivity and ceramide content in human skeletal muscles. To do so, insulin sensitivity was quantified in 27 healthy male using euglycemic hyperinsulinic clamp technique. Insulin sensitivity was compared before and after an experimental treatment in which an experiment group infused with lipids. Vastus lateralis biopsies were taken before the clamp was performed and ceramide content was measured. The results from the euglycemic clamp were related to total muscle ceramide content in each individual. The following figure depicts the relationship between muscle ceramide content and insulin sensitivity. As can be seen, as ceramide content increases, insulin sensitivity, which is denoted by the M value, decreases. The researchers attributed this impairment to the inhibition of AKT signaling. In doing so, they cited a study conducted by Summers and colleagues which demonstrated that ceramides reduce AKT activation in the presence of insulin. Such findings confirm our hypothesis that increased ceramide production by a high-fat diet impairs insulin-mediated AKT phosphorylation.
To recap, the mass that we are considering in this problem is GLUT4 content, and as Drew explained, we believe that GLUT4 content was going to be less in a person with a high-fat diet because the inflow, so the translocation of the GLUT4 vesicles to the skeletal muscle membrane would be reduced. Now, why did we think this? Well, we knew that high free fatty acid diet was going to mean high free fatty acid in the blood. And if we have more high free fatty acid in the blood, we're going to get more fatty acyl-CoA in the muscle, leading to increased ceramide, or sphingolipid formation. As we explored, more ceramides means more deactivation or inhibition of AKT, which is a key player in the insulin-mediated pathway, which leads to the translocation of GLUT4 vesicles to the skeletal muscle membrane. As we saw in the study that John presented, if we have more ceramides, we're going to have less insulin sensitivity. And the study said that this was because of AKT, or PKB. So, our hypothesis looks good. More ceramides, more inhibition of pathways that lead to insulin-mediated transport of GLUT4 vesicles to the skeletal muscle membrane, less inflow to the GLUT4 pool, which means less GLUT4 on the skeletal muscle membrane.